question is from Bablu Gupta, which says, can all microproteins work? No, I mean, if your way of identifying microprotein is really sure, they should all work. Although I would always suggest that the biology is science of uncertainties. So you should always test everything experimentally before predicting anything or going through. So for example, we screen a lot of things and then we funnel down into limited stuff, which is really confident one. They all should work fine, providing that you validated everything beforehand. I hope that answers your question. So next question is by Asmita. Can microprotein play a role in detecting viralities? Of course, this is really new area which hasn't been explored very well. And I would suggest everyone should go through the papers Stephen is publishing or I have shown in my slides. There are certainly roles associated with the pathogenicity for microproteins. Only problem is there are not too many labs in world those are working with microproteins, and the resource is there, information is there, not too many people to play with. Not everyone can go and work with the, if we are working with flowering time, you can't go and work with the viral diseases. So sure, you should go and read more about it. Next is with the Juma Datta. Can transcription factors be considered as microproteins? No, transcription factors, if they have been through the certain things, for example, alternative splicing, and they lost a domain and retain a protein-protein interaction domain. So how things work is transcription factor have two domains generally, typically, protein-protein interaction domain and a DNA binding domain. Imagine if situation is there where during alternative splicing or polymorphisation site or by processing a protease, the DNA binding domain got depleted or deleted, and the domain left is protein-protein interaction domain. It is the case that I showed with the transcription factor studies I showed before, for example, MIP1A or ZIPR3. The protein interaction domain was there, but DNA interaction domain was lost. So that is the case where transcription factor are being targeted by microproteins. Yeah. So what are the tools to identify microproteins in animal cells? Pipeline designed by Daniel is applicable. Yes, it is applicable. Even the new pipeline I have showed recently in G3. Any genome, you know, there are, even in Daniel's paper, there are a number of microproteins reported already from mice as well as human. And in fact, he also mentioned something related to cancer. This is another huge part. So number of labs, they are identifying small regulatory proteins by calling them small proteins or small ORFs. And there are a lot of cases, such cases in cancer, cancer situation where proteins or ORFs are undergoing alternative splicing or other things, and then forming a smaller version of protein, and they could be potential microproteins. So Sanjana, how to identify microprotein? I already mentioned a number of papers, but if you still want to go through, I would suggest if there is an organism that we have already studied, I would say just go through the paper in supplementary file. For example, in recently published G3, there is a supplementary file, identify microprotein from these 6 genomes that we studied in plants. So you can directly Google, not Google, I mean directly search for your protein of interest there in the sheet we published. Are there identified microprotein? I mean, they are not identified and validated experimentally, but the recent paper in G3, it suggests a huge implication of microprotein. Go through paper and you will find a lot of interesting things, especially in plants. Yes, you can, Dr. Satyapal Singh, yeah, you can diagnose. So what I propose sometime, I mean, I also have good belief about that. During disease situation, certainly there are microproteins are being formed. If one could identify them, you can use them as a marker to diagnose the disease. The only factor is, again, experiments. validate experimentally, and then once you have a good candidate, you can always use that as a marker to diagnose the disease. Kostova, do microproteins involved in silencing of genes? If yes, what is mechanism? So I already told the difference between post-transcriptional and translational. So silencing of gene is actually silencing of mRNA that happens post-transcriptional using small RNA, miRNA, or artificial miRNA. Microproteins target proteins. So post-translationally, not silencing of genes, sorry. Neha Tiwari, are microproteins present in money plant? Why they increase very rapidly? Awesome question. I, I, I wouldn't be surprised. They grow because they have physiological way of growing this way. It's just very naive to say anything about it. But yeah, if you know if the money plant has been sequenced, why not you go through the genome and apply MIFFinder pipeline to identify if there are microproteins associated with this quick growth of money plant. The only scientific way to do that or validate that. So more questions. Dr. Satyapal, again, can we diagnose the disease of plant? Yes, as I mentioned before, identifying microproteins expressing specifically during disease condition and then validating experimentally, that validated microprotein could easily be used as a marker. I mean, you can develop antibodies against it, do ELISA very easily, and through the crushed sample that you prepare from disease plant. I mean, it's a way to, what I think it's a way to develop something important or interesting in future. Harishankar, is microprotein applicable in breeding program such as paddy? Yes, of course. That's why we published this G3 paper, because we want breeders to get involved. And so I'm sure Stephen has a lot of things going on with the breeding companies in future. I mean, in talk, but certainly things will appear in future. So if you are working with the paddy, I would suggest you should look at the microprotein list we published in recent paper. And if you have certain candidates, please identify your candidates, and I would be happy to help you taking them further. So Kostov, do microproteins involve in silencing? Okay, no, I already answered that. Can we develop a microprotein modified plant that may be used to reduce contaminants? So I showed that there is a case where microprotein control ion channel. If you know certain ion channel, those who took up soil, took up contaminants from soil through root, why not? You can design a microprotein, either naturally identify that or synthesize one using the approach mentioned in plant-phase paper, and then target your ion channel or a transporter, for example, using microprotein, overexpress that, and then test your plant over the contaminant soil. You could see something. I mean, again, you can talk a lot of hypotheses, a lot of ideas over plant biology and experimental biology, but again, everything funnels down to how concrete experiments you design to validate something. So Ibrahim, microprotein often set to as a decoy, does interaction with the target result in reduced activity? What is the truth? Yes, this is a quality question, Ibrahim. Thank you. So decoy concept is pretty much similar to microprotein. They reduce activity, and it is actually true. I would suggest you to visit Joshua lab in Yale. He was colleague of Stephen, and they both did postdoc together. And Joshua moved into decoy part, and Stephen started microprotein. So there is an overlap between these two concepts, protein decoy and microprotein regulation. And they both are interesting, I would say. So what are application of microprotein in synthesis of secondary metabolism? Shani has this question. So the application of microprotein is in metabolic engineering, and evidence will come soon in future from Stephen's lab. And I am sure if you go through the list of microprotein we published, you will find number of microproteins targeting metabolic engineering, metabolic pathway proteins, especially the key enzymes and the step regulators, and those proteins could be explored very well by community. For example, if you want to take one of most confident protein from our list, you are welcome, and please come back. I would happy to help, or you can always write to Stephen. He's really good replying and helping people. I would second my answer that I answered to Rajni about reducing contaminants. Metal stress can be combated very easily, but again, it's a pipeline that I showed on last slide and somewhere in between, how to identify and validate microproteins. There are a lot of microproteins unexplored, and this is what inspired me to do this presentation. Instead, I left the lab, but I always feel close to this topic. And yeah, there are opportunities there in regulating metal stress in plant. So Shanmugam, what is average size of MIP? Average size, I would say the pipeline we propose is 140 amino acid to 150 amino acid, not longer than that. No, base pair actually, why I'm saying amino acid, okay. Yeah, I propose, I shows that in slide. Microproteins can combat, yeah, she replied, I replied her. What are consequences of overexpressing? What are consequences of overexpression and loss of action of microprotein? So consequences are, for example, they all depends on which microprotein we are talking about. In case of zippers, which I showed the first case earlier, it says if you overexpress and then you knock out, there are different ways of leaf phenotype. For MIP transcription factors, if you knock out both MIP1A, MIP1B, there is no phenotype as published in paper. And if you overexpress MIP1A, there is a delay in phenotype. Can we identify microprotein in M? Oh, virus genomes are really small, and I can't comment on it. What is happening to virus genome after entering to host? It's really uncertain. It's really complicated to answer this question, but I would suggest if you go through paper, you might find a number of things to address, especially with the viruses and pathogens.
Thank you, Kaushal.